Unlocking human genetics, empowering knowledge, unravel the mysteries of inheritance. Welcome to the fascinating world of genetics. Genetics, a branch of biology, deals with the study of inheritance and variations. It is the key to understanding who we are, why we are unique, and how we inherit characters from our parents. Hi students, I am Sulekha, Faculty of Botany, Professor P.C. Thomas classes and Chaithanya classes Thrissur. Gregor Mendel, the father of genetics, conducted pioneering experiments on pea plants, that is Pisum sativum, in the mid-19th century, discovering the laws of inheritance and laying the foundation for modern genetics. Genetics, as we know today, would not be complete without mentioning the pioneering contributions of the following scientists who have been instrumental in illuminating the intricacies of genetics. Key discoveries in genetics start with the foundation work of Mendel in 1865, Walter Fleming's discovery and naming of chromosomes in 1882, Walter Sutton's chromosomal theory of inheritance in 1902, and later Frederick Griffith's experiment in 1928 showed bacterial transformation, paving the way for understanding DNA's role as genetic material. Oswald Everett, along with MacLeod and McCarthy, are credited with identifying DNA as the genetic material through their landmark experiment, which was later confirmed by Alfred Hershey and Martha Chase in 1952. The pioneering work of Maurice Wilkins provided X-ray diffraction data of DNA, Rosalind Franklin's X-ray crystallographic studies of DNA images, and Erwin Chargaff's base pairing role of nucleotide of DNA paved the way for Watson and Crick to propose DNA's double helix structure in 1953. Matthew Michelson and Franklin Stoll in 1958 experimentally proved that DNA replicates by semi-conservative method. These groundbreaking achievements earned Watson, Crick, and Wilkins the Nobel Prize in Physiology and Medicine in 1962. Rosalind Franklin passed away in 1958 before the Nobel Prize was awarded. Watson and Crick's unraveling of DNA's double helix structure in 1953 collectively revolutionized our understanding of genetics. Now let us explore how DNA is organized into genes and chromosomes. DNA, or deoxyribonucleic acid, is the genetic blueprint for life. Within this blueprint, specific sequences of DNA are called genes. Genes contain instructions for synthesizing proteins, controlling traits and characteristics. DNA's genetic code shapes an individual's unique identity. Genes transcribe messenger RNA, that is mRNA. The process is called a transcription and carries instruction for the synthesis of a particular type of protein. During protein synthesis, the process is called translation. Ribosomes read mRNA's code by linking the amino acids in precise sequence, forming polypeptide, which later fold into functional proteins. Specifically, the order of nucleotides in a gene determines the order of amino acids in proteins. All proteins are coded for by genes found in the DNA of the cell. Proteins as enzymes serve as the master architects of character expression, designing and refining traits through precise biochemical reactions and metabolic pathways. Finally, proteins as enzymes regulate gene expression, and thus DNA's double helix structure holds life's secret. Now we will unravel the fascinating process of DNA packaging. DNA wraps around histone proteins and coils into chromatin fibers, which is the coiled DNA protein complex. During cell division, after DNA replication, chromatin condenses and forms visible rod-shaped structure is called a chromosome. This intricate beauty of DNA packaging is a wonder of life itself, and this process allows DNA to fit within the chromosome of cell nucleus. Stretching 2.2 meter long DNA is intricately condensed into 46 unique chromosomes of varying sizes through chromatin formation and chromosome condensation, which enables life's remarkable diversity. Let us learn about how chromosomes are structured and organized. Humans have 46 chromosomes arranged in 23 pairs and are categorized into autosomes and sex chromosomes. Sex chromosomes determine an individual's sex. Females have 2 X chromosomes with the XX configuration, while males have 1 X and 1 Y chromosome with the XY configuration. But how does this relate to baby's sex? It is determined by sperm. Sperm can carry either X or Y chromosomes. The type of sperm that fertilizes the egg decides whether the baby will be a boy or girl. Typically, humans have 46 chromosomes, but variations in chromosome number, often due to mutations or non-disjunction during gametogenesis, can lead to genetic disorders. Karyotyping is a diagnostic tool that visually displays an individual's chromosomes arranged by size and centromere position, helps to identify chromosomal abnormalities that leads to genetic disorders. Due to complexity and breadth of this topic, we will be splitting this video, and in our next video we will discuss genetic disorders in depth, discussing their types, symptoms and inheritance pattern. Thank you for watching, and join us next time for more insights into the fascinating world of genetics.